We have Phineas Gordon and Bella Martinez, who are representing the Discovery Lab, who also focuses on antibiotics. So you all are kind of warmed up for this second talk. You can take over whenever you're ready.
Hi, so like she mentioned, I'm Bella Martinez and this is Phineas Gordon, and we're here to talk to you about screening for antimicrobial compounds in Salmonella typhimurium. And again, like she said, we're from the Antibiotic Drug Discovery Lab. So what are antibiotics? Antibiotics kill or inhibit the growth of bacteria in a person or animal. And what they do is they exploit the cell wall, DNA and RNA synthesis, folate synthesis, the cell membrane, and protein synthesis. So some of the biggest misconceptions of antibiotics are, one, I don't want to take antibiotics because I will develop antibiotic resistance. This is not the case. You yourself do not develop antibiotic resistance. The bacteria cells inside of you can, and that's what happens, but you yourself as an individual do not develop that resistance. Another one is antibiotics are the solution to any type of sickness. This is not true, and this is actually a contributor to antibiotic resistance. Doctors will prescribe antibiotics to illnesses, and it's not fitting. In addition to this, we have antibiotics cure viruses. This is, again, not true. They are for bacterial infections, not to cure viruses. So some other causes of resistance are the overuse and misuse of antibiotics. As you can see in these figures over here, the antibiotics sold in the US, 81% of them are for livestock, where 19% of them are for humans. And the thing is farmers aren't even using these antibiotics necessarily to treat illnesses and bacterial infections in their animals. They're actually using them to promote growth and make it easier for them to get fatter and then sell more meats. In addition to this, we have the misuse, and this happens in hospitals where superbugs like MRSA develop due to the constant use of disinfectants. So how do we solve this problem? From this figure, you can see that there have been no new classes of antibiotics discovered since the 1980s, which means there have been decades where there's a discovery void. So the strategies of antibiotics are limited. As we've mentioned, they're overused and bacteria are becoming resistant, and no new classes have been discovered for the past decades. So how does antibiotic resistance actually occur? As you can see in panel 1, there are a lot of bacterial cells, and the purple ones do not have antibiotic resistant DNA, but the pink ones do. So in panel 2, when antibiotics are introduced, the purple ones that are not antibiotic resistant, they die, and the ones that are pink survive. And then in panel 3, you see that they're able to repopulate and take over the population. And then in panel 4, you see that they can actually share their DNA with other bacteria, which just causes more resistance.
So how do we determine if something is a possible antibiotic in nature? So for example, we could find something in nature that shows a decrease in bacterial growth. So for example, honey has been shown to have a decrease in bacteria growth. So what we can do is that we can take our honey and we find a compound within there and we extract it out. And then after that, we then test it in a lab where we take a 90 well plate and then we place it in there with our salmonella. And then what we do is that we incubate them for about 24 hours. And then afterwards, then we look at it under a spectrophotometer, which basically shoots light straight at the well plate and then reads the absorbance of the bacteria itself or in the individual wells. So how we can decode the analysis is that on the higher side of the absorbance would be bacterial growth. So a higher number means that more bacteria grew and your compound didn't work. But on the lower end, there would be a decrease in the amount of bacteria because the absorbance number is lower. And so that could indicate a possible antibiotic. And so after crunching our numbers, then we look at if it is an inhibitor, which means that it's an antibiotic, or if it's an inhibitor and an antibiotic, or if it's a nutrient. And then so then we rinse and repeat this experiment over and over and over. So we are given a giant library of like 10,000 possible compounds. And then we experiment over and over and over, rinse and repeat, and then we isolate it down to about 250 compounds which show antimicrobial properties. And then after that, then we put it into clinical testing where we put it in rats, humans, etc. And then after all that, then we find which ones are toxic to humans or have the least amount of side effects. And then after all the years of research and all the funding, then we get at least one statistically. So this is what our graph would look like in our experiment. So it's a standard bell curve with our different standard deviations. And at the top, so this would be the response to bacteria when they're exposed to these different chemicals. So DMSO is a neutral chemical where we just use it to put the compound in to dissolve. And so the DMSO would sit here up at the top, so it would represent our negative control. And then on the far left, we would find our ampicillin. This is what is like a registered antibiotic from the FDA. And so it's below this 2 standard deviations. And underneath here are the responses to the antibiotic. And over to the right, you would find something like agar, which is a nutrient which would stimulate the growth of bacteria. And so what we're trying to find is our possible compounds below the 2nd standard deviation over here on the left.
All right, so why do we choose to study Salmonella typhimurium? This is because it's a great model organism. It's safe. There's no risk of getting typhoid fever in the lab. And this is because Salmonella typhimurium is not the same as Salmonella typhi. Salmonella typhi is responsible for typhoid fever. However, Salmonella typhimurium displays symptoms of typhoid fever in mice, but just food poisoning in humans. In addition to this, it's very cheap and accessible. And again, like I mentioned before, biologically, they're very similar. Okay, so here we can see a little bit better Salmonella typhimurium versus Salmonella typhi. As you can see, everything is the same. The green, the orange, and then the DNA in here. There is just extra antigens on S. typhi. So it makes it a great model organism for us to test against.
And so in a clinical setting, this would look very different, obviously. As Bella said, S. typhi causes typhoid fever, which is has been found to have antibiotic resistant properties recently, with new cases showing up every year. Except Salmonella typhimurium clinically just gives you food poisoning. And it's the salmonella you typically think of when people say, oh, don't eat that has salmonella in it because of like rotten eggs or spoiled eggs. But this doesn't cause nearly as many deaths, and the majority of the deaths are in people who are immunocompromised, who have something like HIV, but typhoid fever alone causes most of the deaths, as per year it causes about 4,300 deaths, or 433,000 deaths per year internationally.
All right, so our solution personally that our project focused on was called melittin. It's a polypeptide chain that's found in honeybee venom, but it's not the venom itself. So the reason that we chose this, it was already tested and proven to have possible antimicrobial properties. In addition to this, before, venoms have been increasingly promising in vitro studies. So specifically in the years past in our discovery lab, wasp venom, blue scorpion venom, and snake venom have all been tested. However, due to their high cytotoxicity, it made them unsuccessful. And so the reason why we chose to take a compound out of venom itself was because of this, because the cytotoxicity of melittin is a lot lower than if you tested actual honeybee venom.
So here is, after running all the experiments, here's what we found in our results. So shockingly, we did find that ours was a statistical hit, as it falls below this red line, or those 2 standard deviations we were talking about earlier. And you can see on the far right where ampicillin would fall. And so it shows very similar properties where it kills the bacteria itself. And so here's a closer image of that, where we find that at these two concentrations, we were able to have antimicrobial properties in melatonin. And so here's another image of our concentrations. And here's DMSO on the right, which is supposed to be like our negative control. And so this wasn't supposed to have a positive or negative effect on our salmonella. So this is very promising research. As we can see, it's very antimicrobial. And so it could be possible to use or test it as an antibiotic in the future.
So our research has real life implications. Our group specifically wasn't the only one to have found working compounds. There are other groups that were in our class that also worked and identified statistical hits. The research that we completed is a great step in the right direction for discovering new antibiotics and filling that discovery void. We'd also like to acknowledge Dr. Pamela Harvey for helping us with this entire research project, specifically our group. We'd also like to thank Dr. Corey Detweiler and Detweiler Lab for advocating for our research. We'd also like to thank all of the incredible TAs and give a shout out to our own TAs, Daniel Hassel, Anila Narayana, and shout out to Cheerska. And finally, we'd like to thank Lee Neiswander and the Department of Molecular, Cellular, and Developmental Biology here at University of Colorado Boulder. Thank you, and we're open for questions now.
I have a question. I was wondering so you showed that melatonin has really nice antimicrobial activity in Salmonella typhimurium in your test where you culture Salmonella in liquid media. What would be some of the things that you would do if you could like stay in the class for another year? Like, can you think of like what kinds of things would you have to do to move this closer to clinical trials?
Well, in our graph, we saw that there were two different concentrations. There was a large jump from one of our concentrations to another a little bit higher. So what we would want to find is a concentration that's just right. And so it doesn't just exemplify toxic traits within people or mice even. So what we would want to do is that we would want to find something that is similar, or sorry, we'd want to find something that would have the most amount or the highest punch with the least amount of concentration. And so we would also want to find out if it is bacteriostatic or bactericidal, which means that if it was bacteriostatic, it would mean that it would just inhibit something about the bacteria. And so it would share more similar properties of an antibiotic. But if it was bactericidal, then that means it would just eliminate the bacteria. And so that could possibly mean that it would eliminate even a human cell.
Yeah, and I think what I would do is I would just run the whole test again. So we only actually did one trial, which is why the data looks a little bit interesting for our positive control. But I would rerun the whole trial just to see like, okay, is this valid? Like, is there really like, is this really like actually a antimicrobial possibility? So that's what I would do.
Great. Christian has a question in the comments, and I feel like it's a really good question. The structure of your compound is enormous. This is a very, very big compound. Have you guys given any thought about whether or not that would be an issue moving the compound forward?
So this is a great question. We have not considered this in our research. It was very preliminary. The structure itself is much larger because it's an amino acid. So honestly, more testing would have to be done to determine if there was any like specific implications of it being such a large compound. So for classes who are going to take our research and continue it, we will let them know to keep that in consideration.
We have time for one last question. I'm going to pull it from the comments from Lewis, who is asking about the physiological relevance, basically, of the concentration that you tested. And I think what he's asking is, how does the concentration of the compound that you tested compare to what you can get into a person, for example? Is it relevant?
Yeah, no, that is extremely relevant. So the max dose in the human bloodstream is 10 micromolar. So if you remember from the graphs that we had, it worked at 6.25 micromolar and then again at 12.5. And now the reason for showing both of those is obviously like once you get past 10, like it's going to become more toxic in the bloodstream. So like Phineas mentioned before, we would have to narrow down our research and find a really good concentration where it still works, but again, isn't going to kill you. So yes, the concentration does matter because we want something that is a decent concentration without it losing those properties of the antimicrobial properties.